Welcome to Nursing Lecture Within Class. In today's video, we will discuss about the erythrocyte, or red blood cell. Human red blood cell are non-nucleated circular disc-like cell present in the blood with a mean diameter of 7.5 micrometer. The red blood color is due to the presence of pigment hemoglobin. It is interwoven in the stroma of red blood cells so that even if an RBC is cut, there is no extrusion of hemoglobin. The volume of a single RBC is about 90 micrometer and surface area is 120 micrometer square. Normal shape of red blood cell. Normally the red blood cell are disc-shaped and biconcave. The central portion is thinner and periphery is thicker. The biconcave counter of red blood cell has some mechanical and functional advantages. Advantages of biconcave shape: it increases the surface area of red blood cell for the diffusion of respiratory gases. Because of the biconcavity, red blood cell can squeeze itself through the capillaries, which have smaller diameter than that of red blood cell. If you're talking about the composition of red blood cell, so red blood cell is composed of water and solid content. Water is 60 to 63%, while solid content is 73, else 37 to 40%. 34% of solids is hemoglobin, and the rest 3 to 6% is constituted by the other protein, potassium, iron, phospholipids, cholesterol, adenosine triphosphate, ATP, mainly oxidative enzyme and coenzyme etc. If we're talking about the anatomical structure of red blood cell, so red blood cell is bound by a membrane made of a double layer of lipid and protein layer in between. It is a selective permeable with specific protein channels, pumps, and gates. Lipid layer consists of phospholipids, cholesterol, and small amount of glycolipids. The glycolipids contain oligosaccharide sequence on the outer surface which form ABO blood group substance. The peripheral and integral membrane protein of red blood cell provides structure integrity, for example spectrin, actin, tropomyosin, glycophorin, ankyrin, etc. Spectrin is the important protein molecule which determines the structure integrity of red blood cell. These proteins form the skeletal of red blood cell, which provide its ability to change the shape as it passes through narrow capillaries and to resume the biconcave shape afterward. Mature red blood cells do not have a nucleus and lack important cell organelles like mitochondria, so it cannot divide or synthesize protein. Red blood cells mainly contain hemoglobin that depend entirely on glucose metabolism for energy supply. If you're talking about the normal range and normal value, normal range is 4.5 to 5.5 million per mm in male, while in female this ratio is slightly lower, from 4 to 5 million per mm 3 in female. RBC are counted on a special chamber called new birth counting chamber. If we're talking about the erythropoiesis process of red blood cell or red blood cell genesis, first of all, pluripotent stem cell. After multiplication, another cell form called colony forming unit EO. Then another changes occur in the cells, which is called free erythroblasts. Then free erythroblasts convert into normoblasts. Normoblasts convert by 3 stages: early, intermediate, and late. After late stage is convert to reticulocyte, and reticulocyte then convert to mature red blood cell, which is called erythrocyte. If we're talking about the properties of red blood cell, first property is relux formation. When blood is taken out of the blood vessel, the red blood cell fire up one above another like the fires of point. This property of red blood cell is called relux formation. It is accelerated by a plasma protein namely globulin and fibrinogen. We're talking about the specific gravity of red blood cell, which is 1.0922 to 1.101. Suspension stability is the another properties of red blood cell. What is suspension stability? During circulation, the red blood cell remains suspended and dispersed uniformly in the blood. This property of red blood cell is called suspension stability. What are the lifespan of red blood cell? Average lifespan of red blood cell is about 120 days after the lifetime. The senile or red blood cells are destroyed in the reticuloendothelial system (RES). If you're talking about fate of red blood cell, when the RBC become older, after 120 days, the cell membrane become very fragile, so these cells are destroyed while trying to squeeze through the capillaries, which have looser or equal diameter is that of red blood cell. The destruction occurs mainly in the capillaries of spleen because the capillaries are very much narrow, so the spleen is called graveyard of red blood cell. The destroyed red blood cells are fragmented and HB is released from the fragmented parts. HB is degraded into iron, globin, and porphyrin. Iron combined with a protein called apoferritin to form ferritin, which is stored in the body and reused later. Globin entered the protein it default for later uses. The porphyrin is degraded into bilirubin, which is excreted by liver through bile. Daily 10% of senile red blood cells are destroyed in normal young healthy adult. It causes release of about 0.6 gram per dl of HB into plasma. From this 0.9 ng per dl to 1.5 ng per dl bilirubin is formed. Next discussion will be the function or physiological role of red blood cell. Number first, transport of O2 from the lungs to the tissue. It be combined with O2 to form oxyhemoglobin. Number two, function transport carbon dioxide from the tissue to the lungs. It be combined with CO2 and form carboaminohemoglobin. Number third, function is buffering oxygen in the blood. Its function is a good buffer. By this action, it regulates the hydrogen ion concentration and thereby play a role in the maintenance of acid-base balance. Number 4, function in blood group determination. RBC carry blood group antigen like A antigen, B antigen, and Rh vector. This helps in determination of blood group and enable to prevent the rejection due to incompatibility during blood transfusion. Thank you for watching this video. For more video, please visit Nursing Lecture Within Class. Thank you.